Global climate change is happening, so is the global population increase. It is a huge challenge to feed 9.8 billion people as predicted in 2050, especially with crop production in much warmer and water-limited environments. The key for crops to survive is to have better use of water. We breathe, so do plants. The mouths of plants are called stomata. These cell structures on leaf surfaces form pores that allow CO2 to enter a leaf where photosynthesis takes place. However, simultaneously water vapor escapes through the same pores as a byproduct. The loss of water vapor accounts for more than 95% of water uptake by plant roots. Thus, we want to make plants cleverer in terms of water usage by reducing stomata density and size, so that plants like corn, which is a species I'm studying, can have access to just the right amount of CO2 to do highest photosynthesis. Meanwhile, water loss reaches to minimum. The very first step to approach to it is to know how many stomata there are and how big they are in each corn plant. The left side of the background image shows how corn leaf surfaces look like. The size of the image is about 1/10 of your fingernail. Those round-shaped cells with a pore in the center are stomata. And imagine I have to count all these cells one by one on thousands of images like this. It is like counting a handful of sand. That is no fun and might reward me a pair of cross eyes after 5 years of my PhD program. What is worse is to measure the size for each stoma. That is almost impossible to be finished in a large-scale experiment. Good news is that I didn't just spend my time counting and measuring, but rather I have developed a machine learning model, offering a way more efficient solution. Basically, I can train the algorithm by feeding it the contours of cells recognized by me. The computer extracts and learns the features of the cells, and after dozens of training cycles, the machine gains the ability to recognize and highlight these cells, as you can see on the right side. Summarization of stomata density and size measurements are done within seconds. It breaks the bottleneck of the time investment in data collection for stomata characteristics. With the tool in hand, I can extend my research to hunt for genes regulating stomata patterning. We will be able to modify these genes to test our hypothesis and breeding for corns with less water transpiration. My goal is that in the future, farmers will be benefited of having corn plants that are more drought tolerant and use less water.